everyone know. Welcome to this, the second EBR seminar. And maybe some of you are not familiar with this concept of evidence-based research or EBR. So shortly, it is a concept that promotes a systematic and transparent approach considering earlier studies and the end user's perspectives when you are planning, justifying, designing a new study, and even when you are interpreting the new results you are having. I can tell you that yesterday in the first seminar we had, we were presented the EBR concept in a little more details. We were sharing the accomplishment of our cost action for the last 4.5 years related to EBR. And we also presented the EBRNetwork. So just for everyone to know, you can go to EBRNetwork.org. And here you can get further information and you can even sign up for our newsletter. I am Hans Lund. I'm professor at the Western Norway University of Applied Sciences and a chair of the EBRNetwork. So the interesting thing here is, and that's really exciting, I'm really looking forward to this, we are going to discuss the concept of evidence-based research and social science. And our approach will be based on the question, replication in social science, too much and or too little. So please use the Q&A option for questions or comments during both the presentations and the panel discussion. We will read them and share them with the speakers and the panelists. So our first speaker is Vivian Wong from Collaboratory Replication Lab, University of Virginia in United States. And her title is Moving from What Works to What Replicates, Designing Systematic Replication Studies. Please, Vivian, the floor is yours.
Hello, everybody. Thank you so much for inviting me to speak. I'm excited about giving this presentation. It's very early in the morning, so there's lots of coming and going with my kids and the dogs this morning. So sorry about the distraction. So I am really excited to hear your questions, your comments. When we had a pre-meeting, Vivian suggested that it was okay to be provocative. So you should feel free to, I'm also comfortable presenting to economists, so you should feel free to interrupt and ask questions or that kind of thing. I enjoy having interaction during the talk. So with that, I'll go ahead and begin. I'm going to share my screen, if that's okay. Sorry, I have some interactions happening back here with my dog. Okay, is everybody able to see the slide? That's good. Okay, great. And then again, the host should feel free to signal me if I start to run over on time. So feel free to give me signals to wrap it up. Okay, so what I'm going to do today is I'm going to talk a little bit about some of my work in collaboration with Peter Steiner at University of Maryland, and talk a little bit about our methodology work for conducting high quality systematic replication studies, and an idea for how systematic replication studies could be used for the purposes of generalizability. This work is supported by 2 methodology grants from the Institute of Education Sciences. I'm going to spend the bulk of my talk today talking about replication and what it is and the stringent assumptions that are often required for replication. And my hope is that by defining replication formally, it will shed light on some methodological issues related to both replication and generalizability. What I'm going to do is I'm going to sort of, because we're short on time, I'm just going to say basically what I'm going to try to argue today or convince you of, or you should feel free to challenge me of. So first, what I'm going to argue is that the direct replication of results to be achieved, the assumptions or the requirements for replication are going to be quite stringent, as you'll see. So perhaps we should not be surprised about some of the low replication rates that we do see in lots of replication efforts in the area of social sciences. And then second, what I'm going to argue is that by understanding assumptions required for replication, we can see how we can derive replication designs to test systematic sources of effect variation, and that these replication designs in the social sciences are feasible, ethical, and often desirable in field settings. And then what I'll also argue is that methodological issues related to replication and generalizability require design-based approaches for thinking prospectively, not just about planning a single study, but a whole series of studies that you might use in, for example, a meta-analysis. Okay, so as a methodologist, replication is one of the few areas of work that does not require much formal motivation or introduction. And the reason why replication of results has garnered so much public attention is because at its core, it asks about the trustworthiness of scientific results. And the idea here is that we can't ask the public to trust us for producing scientific evidence for decision-making if we don't have confidence that our results are not valid, replicable, or generalizable to some target population of interest. And some of this anxiety comes from well-publicized efforts from scholars such as Brian Nosek and the Center for Open Science. Their initial replication efforts concluded that the replicability of results in psychology, as well as other areas, have been surprisingly low. And so the response to what some have called the replication crisis has been to establish incentives for promoting the replicability of results and to develop a culture in which the scientific community replicates results before recommending for decision-making purposes. So for example, the National Institutes of Health supports the training of researchers and graduate students to promote the replication of results. And there have been some suggestions that results should be replicated prior to publication or to be included in evidence registries such as the Works Clearinghouse. However, the trouble with this is that replication as a method is not yet well established. We don't have strong consensus on what replication is, what constitutes as a replication studies, and how to analyze and interpret one. And for sponsors and funders of replication efforts, it's hard to often know what a high quality replication study even is. So the purpose of our group is to examine sort of these methodological questions on what is replication and how to conduct replication studies in field settings. Our group is organized around something that we call the causal replication framework, which provides a common approach for understanding what replication is, why it might occur, and requirements for high quality replication. And our central ethos is that replication itself, or replication failure itself, is not inherently a problem for science, as long as we have systematic ways for understanding why failure occurred across studies. And another way to think about replication failure is to think about it as identifying sources of effect heterogeneity, which is going to be central for being able to generalize results to samples and contexts that are not observed. So let's go ahead and begin and dive into what we mean when we're saying by replication. So I'm going to begin by talking about a definition from Schmidt, which is what I see is the most sort of commonly cited definition for replication. And Schmidt describes replication as a methodological tool based on the repetition of procedure that is involved in establishing a fact, truth, or piece of knowledge. Now, this definition doesn't seem uncontroversial. It's similar to sort of definitions that people give when they're sort of asked ad hoc, what is replication? And when most people read this definition, the part of the definition that many sort of hone in on at the beginning is around the repetition of procedures. This is often what people mean when they say direct replication. It means that they're going to replicate procedures, they're going to replicate protocols and methods as closely as possible to the original study. So here, if replication is defined by close repetition of methods and procedures, then the quality of replication is determined by how closely a study is able to repeat methods and procedures from the original study. However, if you've ever conducted a replication study, you've already recognized the problems with this, right? First, the original study may fail to report all of the relevant necessary methods. And this may not be because a lab or a research team is trying to sort of cover up their methods, but sometimes it's hard to know exactly what to report in, a scientific study. Second, for people who work in field settings, we know that things often go wrong or things deviate from the protocol in field settings. And then the question often comes up, which is, or the state of the art sort of improves. We get better measures for things, we get better methods for things. So the question often arises, well, should we be replicating the same flawed methods and procedures, or should we adopt the new sort of procedures or measures that we know are better from the original study? And then finally, a third question is that there feels like there's an inherent challenge with how we've defined replication, as it's sort of stated here, right? The goal for replication is to be able to uncover some sort of truth or knowledge here, but this standard is all about replicating procedures and methods, which is probably important to thinking about replication, but is not sort of the central goal of what replication is meant to address, okay? So let's look again at this definition, because I actually think it's a pretty good one, but we're going to focus on a different part of the definition, which I've highlighted in blue here. It's the focusing on the fact, truth, or piece of knowledge, okay? And here we're going to think about how do we operationalize this, right? It sounds like it's a lofty goal, but maybe it's too ambiguous. And here what I'm going to draw upon is that one of the major methodological advances in the program evaluation literature over the last 50 years has been the introduction of some notation. Here we call it potential outcomes notation. Basically, this development has allowed us to write down mathematical definitions of what we mean by fact, truth, or piece of knowledge, and to be able to derive different research designs. Mostly the introduction of the potential outcomes framework has formalized our intuition for research designs, and they've also allowed us to be able to start developing procedures for testing sort of assumptions for these research designs, okay? And basically what we're going to do here is we're going to apply this reasoning to think a bit more rigorously about what we mean by replication studies. So to begin with, I'm going to argue that replication is about the replication of a causal effect for a well-defined treatment contrast, which we're going to call a causal estimand. And what I'm going to do is I'm going to show you that there are basically 5 conditions or 5 assumptions that are required for replicating the same causal estimand. And as I talk more about this, we'll see that focusing on the replication of the causal estimand means that replicating the same methods and procedures may help in conducting replication studies, but it's not always required or even desired, okay? But what matters here is introducing systematic variations across studies in order to be able to evaluate the robustness of results, and when results fail to replicate, to be able to understand why that effect was either amplified or dampened, okay? All right, so basically, what does the replication framework say? So in replication evaluation, successful replications of results can only be expected if the causal estimand of 2 or more studies are identical. However, in current practice, the causal estimand is usually not well identified by the original study. And the causal estimand that it's being replicated in replication studies are also not well identified. So under the causal replication framework, the researcher begins their study by clearly defining the causal estimand of interest, okay? And then the assumptions that are required in field settings are appropriate for identifying what this causal estimand is, okay? So this table describes a summary of the assumptions that are required for 2 or more studies in order to be able to replicate results. There are 5 core assumptions for the direct replication of results. They fall into 2 categories of assumptions. The first set of assumptions are what I'll refer to as replication assumptions. They're indicated here by R1 and R2. These assumptions ensure that we're comparing the same causal estimand across different studies. R1 basically means that we're comparing results across different studies that have the same treatment and control conditions and the same outcome measures. And R2 means that the different studies have the same joint distribution of participant and setting characteristics on factors that may amplify or dampen the effect of the intervention. The second set of assumptions that are what I'll refer to as individual study assumptions. They're denoted here by S1 through S3. These assumptions ensure that each individual study in any individual or any replication effort produces a valid estimate of the causal estimate. These are your standard assumptions that are required in any causal study. So for example, S1 requires unbiased identification. This may include that you've used an RCT or a well-implemented quasi-experiment for identifying the treatment effects, that estimates are correctly estimated in an unbiased way, and S3 requires correct reporting, that you've correctly and appropriately reported your estimators and estimates, that there's no p-hacking, that there's no questionable research practices in reporting your estimates. In sort of presenting this framework to different folks, I find that these assumptions are pretty intuitive to folks, right? These are the assumptions that folks would typically come up with if you're sort of thinking about assumptions required for replication. However, they're quite stringent, right? You know that it's challenging in field settings in order to be able to address sort of all of these assumptions. So perhaps we shouldn't be surprised when replication efforts do occur in field settings. Okay. And it turns out that the framework is able to sort of match on to our heuristic understandings for different types of replication designs. So for example, a direct replication study may be understood as examining whether effects replicate for 2 or more studies that have the same causal estimate. So they have the same target population, the same settings, and the same outcomes. And one way that you might introduce systematic variation to test, for example, the reporting assumption S3 is in the reproducibility analyses where you may have the same data, the same syntax file, and the goal is to evaluate whether independent investigators are able to produce and report the same results. Or you might test S1, the identification assumption. Here, you might evaluate whether results from an observational study, such as propensity score matching, are able to replicate results from a gold standard randomized control trial for the same target population. And this type of design is called a within study comparison or design replication studies, because if the results do fail to replicate across the observational arm and the RCT arm, then the researcher might conclude that replication failure occurred because assumptions in the quasi-experimental design were not met in the field setting here. These are examples of direct replication efforts. In field settings, oftentimes researchers are interested in conducting replication or conceptual replication studies, where the goal is to evaluate whether 2 or more studies with potentially different causal estimates produce the same effect. So for example, in order to be able to test whether different implementations of the treatment, S1, for example, treatment stability, is able to replicate the same effect, we have designs for that. We call this a multi-arm treatment design, where individuals might be randomized to different treatment arms. And the goal here is to assess whether the effect is the same across these different treatment arms. Researchers might also be interested in examining whether effects replicate across different subpopulations of individuals. So they might use a blocking design to examine whether effects replicate across different strata of individuals or blocks of individuals or across different sites. So the idea here is that although replication assumptions are quite stringent, we actually have a lot of research designs that we currently use that may be understood as types of replication designs. And when a replication design is well implemented, the idea here is that researchers should be able to discern why the replication failure occurred. Okay. And so that's basically what sort of the first take-home message that I want to argue here is that by prospectively planning and thinking about replication designs, and by introducing and testing systematic sources of variation across study designs. The goal here is to not only look at the replication of results and the robustness of results, but to examine whether these results are replicable across systematic sources of generalization or of heterogeneity. Okay, so how does this relate to generalizability? Well, we find that unlike researchers who come to replication from laboratory or open science perspectives, many field evaluators who are interested in replication are usually interested in this idea of generalization, okay? And so the goal is not just to understand the replicability of effects, but also to understand the generalizability of effects. And usually researchers and decision makers are interested in understanding the generalizability of effects across what folks might refer to as generalizability factors, right? So Cronbach first described what generalizability factors are by presenting his UTOS framework, which include generalizability factors across unit characteristics, treatment, implementations, outcomes, settings, and time. And currently, since I'm talking to folks here who are interested in meta-analysis, what I'll just sort of highlight is that we often think about using meta-analytic approaches for generalizing by combining sort of study results and obtaining some kind of average effect across all of these different studies, okay? However, the challenge here is that meta-analysis involves the synthesis of effects from obtainable study samples, but study samples are recruited for a number of different reasons, right? They might be locationally convenient, logistically feasible, or financially reasonable, okay? And then the challenge is that these study effects are average, but it may not be clear for whom, what, or where these study effects actually represent. So Rubin sort of first made this sort of critique when he asked about meta-analytic results. What is the target population? And even if I could conceptualize it, why should I care about the average effect in it? Now, the meta-analysis will often say is that with enough studies, it's often possible to identify study factors that are correlated with both the size and the direction of the effects through, for example, a meta-regression. Where the goal is to look at moderators that are associated with smaller and larger effects. However, the challenge here is that, again, these are correlations, and it's not always clear how researchers should interpret the associations of effect size with study characteristics or sample characteristics, and whether they can produce valid predictions of the intervention's impact for populations that are not included in the study. So generalization requires some knowledge about the cause of the effect variation or the causal moderators, as well as ability to be able to extrapolate these effects, okay? So what I'm going to sort of very quickly highlight here and encourage folks, and maybe sort of my provocative statement here, is to encourage for folks, especially for those in meta-analysis, to think about what the alternative to meta-analysis, which is response surface modeling here, right? So Rubin suggested response surface modeling as an idea for generalizing effects in '92. Here, I've represented a response surface through what we call an effect grid, for which researchers may be interested in generalizing effects across sort of a surface space, for example, a reading intervention. And here, each cell represents basically a study finding, and the study finding effect is a function of some setting and contextual setting and population characteristics that we might want to generalize over. Now, how is this effect grid formed? Well, this effect grid is formed based on our sort of substantive theory and our hypotheses of what effects are being moderated in advance of sort of conducting the studies. So, for example, in this example that I'm using, the moderators for this reading intervention might include site characteristics, contextual characteristics for how the intervention is delivered, as well as kid characteristics, such as whether or not the kid has a reading disability. And the point here is that what the effect grid sort of highlights is not a single study, but thinking about a constellation of studies and how researchers are contributing a single study in the context of other studies, okay? Once you sort of move into this kind of response surface or effect grid kind of perspective and thinking about a constellation of study effects instead of a single study, it becomes clear that you can start to think about, well, if I was to prospectively design a series of studies, instead of sort of having ad hoc approaches for people just planning sort of whatever study that feels convenient for them, what if we were able to sort of choose an optimal approach for what cell study effects that we would want to observe, and we would want to choose these study effects in a way that are optimal for being able to uncover sort of moderator effects in order to be able to cover basically this grid. And Peter and I have a grant where we explore methods for how to do this. Okay, and then the idea here is that once we select these optimal cells, then teams of researchers would work together and they would go and estimate effects for these specific cells. So they would conduct their own studies and estimate effects for these cells, but then they would also use this to contribute to sort of an overall effect grid. And then the idea here is that once we have some of these estimated effects, we're then able to estimate some of the observed effects as well as uncover some moderator effects here. And once we have the moderator effects, then we're able to then sort of extrapolate and be able to predict to cells that were not able to observe the effects. And that basically is generalization. Okay, so here what I'm sort of arguing is that in order to be able to generalize effects requires a couple of things. It requires both the replication of effects, but also the uncovering of effect heterogeneity in order to be able to sort of extrapolate and generalize effects across, for not a single average effect, but across a response surface. Okay. So, and then how do we think about sort of what this response surface means? Well, basically a flat response surface implies that the intervention works the same way for everyone under every condition. But if there's a lot of sort of variation in effects, like there's like a Craigley kind of response surface, it implies that there is treatment effect variation that varies across sort of different conditions and different population characteristics. Okay, so I'll start to wrap up. I think I'm over time here, but under the causal replication framework, when results fail to replicate in direct replication studies, we may conclude that individual studies are biased or incorrectly reported, but when results fail to replicate in conceptual replication studies, the researcher might conclude that there is a presence of effect heterogeneity, right? And this understanding sources of effect heterogeneity is important because it's critical for generalizing, which is what decision makers often are asking for when they're asking for evidence-based research. We also want to emphasize that that the assumptions for being able to sort of conduct replications are quite stringent. However, there are pathways that we can use if we think about it from an assumption perspective, where we can start to sort of describe and produce diagnostic measures for demonstrating the extent to which these assumptions were met or which they were violated or even systematically varied in studies. So the main message here is that the name of the game, I think, in replication efforts is to be able to understand sources of effect heterogeneity when replication failure occurs. And that the way to do that is through sort of more design-based or prospective designing of studies. Okay, thanks. I'm going to pause for now, and I'm happy to sort of turn it over to Hugh or to answer any questions, however the panelists want to, or the host want to sort of manage it.
Thank you very much, Vivian. Very good. I think that we should wait with the question until we have both presentations, and then we can have good time with the panel and all the participants to ask the questions. There is no question right now in the Q&A. I will really encourage all to use that. So maybe we should go directly to the next speaker, which is Hugh Waddington from London School of Hygiene and Tropical Medicine in United Kingdom. And Hugh's title is What Can Health and Social Scientists Learn from One Another's Replication Efforts to Improve Research Quality? Thank you very much for joining, Hugh, and the floor is yours.
Hi, thank you very much. I'm sorry I was slightly late. Vivian, I caught most of your talk. I'm afraid I'm down with COVID, so I'm slightly out of it. But I'm fine to give the talk. So can you just give me a thumbs up if you can hear me and see my screen okay? Yeah, right, I see your thumbs up. Thank you, Shankar. Okay, great. Okay, so as Shankar just said, this is my talk. I think that a lot of my research, so I'm a development economist by training, but I have a PhD in infectious disease impact evaluation from LSHTM. So a lot of the work I do is cross-disciplinary, and I think there's a lot that we can learn from one another's efforts across disciplines. Just one. So thank you very much. So let me just see if I can move my slides forward. Yeah, okay. So this is really you can see what I'm presenting as kind of an empirical application of a lot of what Vivian just presented, and indeed we were very we drew a lot on Vivian and Peter's work in one of the projects that I'm going to present on. So how can we quantify bias in replication work? Well, there's two main ways of measuring bias. We can measure distance from the benchmark study in internal replication, which, as Vivian indicated, is also called within internal replication is also called a within study comparison. The other main way that we can do it is by measuring the relationship in an external replication. So in other words, that is so whereas an internal replication covers the same population and in a intervention type of study, the same intervention, and an external replication is usually the same intervention but applied to a different population. So two different types of replication studies. But also, as Vivian has covered very well in her research, and I think in that talk, there's actually two main reasons why the results from randomized studies and what I call quasi-experiments, or you might also know them as non-randomized studies of interventions. There's two reasons why those estimates from those studies might differ, and they're not both about bias. One of them is about bias, which is about the obviously about internal validity, and we have I've kind of categorized what I think are the main sources of bias in RCTs versus the main sources of bias in quasi-experiments or non-randomized studies and interventions. So the main sources of bias in RCTs, and the bearing in mind, of course, that the RCT is usually the benchmark study. It's usually considered to provide the unbiased estimator in which we are comparing our quasi-experiments to. And those sources are to relate to subversion of randomization, attrition out, so selection bias out of the study, and what I'm calling motivation bias. That is the effect of people, of study participants being aware that not just that being aware of two things. First of all, that they're taking part in a study and they're being observed, and second of all, an open trial or a unblinded study, that they're aware of which group they have been randomized into. So they're aware if they're either they're in the treatment group or if they're in the control group. In quasi experiments, of course, there are various sources of bias. The main one we often think in a quasi experiment is confounding. So other possible reasons for the changes in outcomes which aren't because of the intervention. But there's also selection bias into the study, and selection of the reported result. So those are the, of course, now there can be problems in both RCTs and quasi experiments across all of these types of bias, but these are the kind of main ones where theoretically we think where the studies are most at risk. Now, but even in the circumstance where we have an unbiased study, whether it's a randomized trial or a quasi experiment, the difference, these two studies of the same populations and the same interventions may still yield different treatment effects, and that's because of this issue called sampling bias, and that's about external validity. And that's basically because of the different treatment effect estimands which are provided in each of these studies. So perhaps the easiest way to explain this is the difference between an intention and between an average a population average treatment effect and a local average treatment effect. And we know that some types of studies, like, for example, instrumental variables and discontinuity designs, give us local average treatment effects, and those local effects may not be the same as the population average treatment effect, even though the local average treatment effect is an unbiased estimator for that particular subset of the population. Okay, so I'm going to present. First of all, I'm going to present some results of a study we did of internal replication studies in development economics. We published this in Evaluation Review last year. This is a systematic review. So here is our Prisma study search flow. We did a fair amount of searching, and we ended up with 8 included studies. And this is this is information about the studies that we included. A lot of them were studies of cash transfer programs, although they weren't solely cash transfer programs. And these internal replication studies used a range of different methods. So 4 of them used some type of discontinuity design, whether they were an RDD, a regression discontinuity design, or a GDD, that's a geographical discontinuity design. And other studies used statistical matching, or even just simply adjusted regression. So that's the OLS, indicated by OLS. And then one study used a range of different measures. So they used a difference in differences, they used instrumental variables, they used matching, and they also used a simple adjusted regression, OLS. What we did was we extracted we calculated 3 or 4 different measures of distance between the benchmark study, which was the RCT, and the quasi experiment, and which I will present in this slide. So we used So what I presented here, we present different additional measures in the paper, but these are the measures that that I want to focus on now. So the first one is the standardized bias. It's just very simply it's very similar to the it's a standardized mean difference where we take the standard deviation in the RCT as the denominator. So that's the absolute distance between the quasi-experiment or non-randomized study of intervention and the RCT. But we also calculated the mean squared error, which is another measure of, and it's probably it's maybe more familiar to those epidemiologists in the room, because there's quite a renowned epidemiologist, Greenland, I think is his name, who he didn't propose this, but he's spoken quite a lot about it. And that's just that's the mean squared error, which is this standardized bias squared plus the variance of the estimate. And it's another way of presenting the difference in the not in the quasi-experimental estimate. And then thirdly, we use this percent of bias removed, and the formula is here, and this was from Vivian and Peter's paper. Okay, so what you're seeing in this table is we used a fixed effect meta-analysis. We did so because these are all internal replication studies, so they're all from the same study settings. And what these studies have done is they've basically they've compared a RCT control group mean to a non-randomized study comparison group mean from the same population. So the same population, meaning that an RCT has been done at a particular time for a particular intervention. At the same time, in some cases, a quasi-experiment was designed at the same time. In a few instances, a quasi-experiment could be undertaken because there had been a household survey that collected the same outcomes data in the same population at the same time. So what we found overall when looking at these 8 studies, of which we found 600 distance measures, we found that essentially, and this was as kind of echoed in Vivian's work and the talk she just presented, it really was the best way to address bias, so was through study design. So in other words, it was I've highlighted the discontinuity designs and the interrupted time series designs here for they're kind of two flip sides of the same study. An interrupted time series is like a discontinuity design in time. The reason why I've highlighted this missing row is because that interrupted time series is not a approach which is typically used by economists for whatever reason. I think I think of it personally, and it's considered to be a credible design in epidemiology and public health, and indeed Donald Campbell considered interrupted time series a very credible design, but there simply aren't any internal replication studies that use the ITS design in development economics, and that's what our review was restricted to. There are these types of studies published in the health literature, and so I would encourage anybody that is so I would encourage those of you that are considering doing a systematic reviews of this literature, you could very usefully zoom in on the interrupted time series and the difference controlled interrupted time series designs, and basically conduct your systematic review similar to the one we have, but focusing on that literature in health. It'd be very useful to know how biased or otherwise ITS designs typically are. But what we found was, for our discontinuity designs, we found almost zero standardized bias, mean squared error of 0, and an estimated percent of bias removed of 95% across 173 estimates. So it's a very credible evidence for the validity of discontinuity designs. And I think it's worth pointing out that some of these studies were prospectively designed. Some of these internal replication studies were prospectively designed. Others were retrospectively designed. And we found almost zero values of bias in each of those instances. And we know that discontinuity designs, they tend to be, they're often designed retrospectively. They don't have to be, but that was an important finding for this literature. And even when we look at some of the other types of quasi experiments where that used what we call selection on observables. So in other words, the control for confounding directly through measured confounders. Even the, for example, matching, we found quite small standardized bias estimators. So for our nearest neighbor matching was the least biased measure, and we still found a standardized bias overall of 0.04 and a mean squared error of 0.07. These are D values. So still fairly small bias measures. The caveat to this finding is that in this paper we also did a assessment. We did a risk of bias assessment for all of the comparisons, and we found they were all at some risk of bias. So that means that either the benchmark RCT, there were some concerns about bias, or that the correspondence, but there was some potential confounding in the correspondence estimate between the RCT and the quasi experiment. So in all of these instances, there were some concerns about bias. What about from external replication? Well, we developed a risk of bias tool around probably about 10 years ago. We developed this tool as a way of, and it was partly based on Cochrane's approaches. This was before Robin's eye had been developed. So we partly based it on Cochrane's risk of bias tool for RCTs. But we believed then, and I still believe now, that when we're critically appraising the risk of bias in studies, whether they're randomized trials or quasi experiments, and whether they've been designed prospectively or retrospectively, the sources of bias, that is the bias domains, are equivalent. So these sources of bias are the same for all types of studies that aim to estimate, all types of statistical quantitative studies that aim to estimate a causal effect. Of course, the difference is that we have different questions in order to measure them. So under for example, for randomized trials, under confounding, we're concerned about the subversion of randomization, for example, which is not, that's not the question we ask for a quasi experiment, which is a study where there simply isn't any randomization. We ask different questions, but the domains of bias, the sources of bias, are the same. So we developed a tool, and this tool was was implemented by a number of author teams under the working with the Campbell Collaboration International Development Coordinating Group. So these are the authors. These are the systematic reviews and meta-analyses that used this approach. They were all published between 2013 and about 2019 and 2019. So we have these 15 or so systematic reviews and meta-analyses that use this tool. So we considered this another way that we could look at the relationship between bias and in RCTs and non-randomized studies or quasi-experiments. But these are, of course, each of these studies is a systematic review, and so therefore it is a study of external replications. They're all systematic reviews of the same or the similar intervention, an intervention that was considered similar enough to be included in a systematic review. But of course, in systematic reviews, we synthesize evidence from interventions from studies which have been implemented in different contexts. So in this case, we're not using fixed effect meta-analysis, we're using random effects meta-analysis. This is just some study descriptives, but what I'm going to move quickly onto it, because in terms of time, is just the results from the meta-analysis. So, excuse me, what we've done in this meta-analysis is we've once again we've calculated a distance metric. So similar to the case of the internal replication studies, we've compared the findings from the non-randomized studies to the benchmark trials. So for every single one of these estimates represents the distance between the pooled effect for the non-randomized studies or quasi-experiments from the pooled effect of the randomized control trial. And this is a type of research which I'm sure many of you will be familiar with. It's called meta-epidemiology. It's basically a meta-meta study. It's a meta-analysis of meta-analyses. And what we found is that when we applied our risk of bias tool for the non-randomized studies that were at low risk, we found, excuse me, for the quasi-experiments or non-randomized studies at high risk, we found these studies overestimated the treatment effect. So you can see that in the bottom here. Whereas when we were looking at the studies with low risk or some concerns, and especially for low risk studies, we weren't finding any difference in the estimated effects. So there's no difference from the benchmark RCTs. Very interestingly, though, when we just look at the RCTs, and we compared the RCTs with, we didn't find any, when we looked at we categorized the RCTs according to their risk of bias. And so in this figure, we're comparing the RCTs with medium risk and high risk to RCTs with low risk of bias. What we found was that actually the RCTs at high risk appeared to have systematically different effects, but the effect size went in the opposite direction. So rather than overestimating the effects, we found that the high-risk RCTs underestimated effects. It's a very interesting finding, and it's quite consistent with a potential source of difference, which is called site selection effects, or sometimes it's called site selection bias. And the principle behind underlying site selection effects in trials is that when RCTs, which of course are prospectively designed, when they're well designed, that means that they are probably the intervention is quite suitable, is designed to be most suitable for the context, so the population of interest, and implementation might be particularly well controlled. So we might expect in those studies for an efficacious treatment, we would expect in those circumstances there to be a bigger treatment effect, and indeed that's what we're finding here. This was just a summary. I'm going to skip over this. Okay. So now I'd just like to talk about the importance of reporting of participant flows. So this is I mentioned at the beginning that I do research where I sit with people that do research on water, sanitation, and hygiene. And the interesting thing about this research is that we have a field. It's a field where randomized control trials and other types of impact evaluations are done on the same topics, but by two different groups of researchers. One of those are the epidemiologists, the others are the economists or development economists. And these and of course RCTs have been done in epidemiology for a very, very long time. RCTs in economics is rather more recent. They've been largely done since about 2000, slightly before. And there are different standards of reporting of these types of RCTs in these fields. So what you can see in this figure is this is the evolution of RCTs in the in environmental health. You can see once consort standards, so that's the reporting of participant flows, was published in 1998, and you see that from about the mid 2000s, when consort was adopted by the top journals, you see that studies started presenting participant flow diagrams in environmental health. And so these days, at least half, if not more, studies in environmental health tend to report their participant flow diagrams. In economics, although at least one journal, that's a journal I work with, the Journal of Development Effectiveness, published some consort standards in 2000. Only two studies of WASH trials have actually published either a participant flow diagram or the information from which that could be reconstructed. Why is this important? Well, it's important if we want to measure key outcomes which are important for the global burden of disease, like mortality. So this slide just shows you a consort diagram. So how this is a consort diagram, which gives you the in case you're not familiar with it, the numbers of study participants by group, together with the reasons for losses to follow up, so the reasons for attrition. And because it's a WASH area, we have losses to follow up due to mortality. But what we've done is we've plotted the 95% confidence interval, and for this particular study, coal. What you can see is that we need quite a lot of estimates in order to estimate a statistically significant effect on mortality. So we're going to need a lot of studies. Well, thankfully, there are a lot of studies. And indeed, this is our results of a meta-analysis we published in PLOS Medicine this year. What you can see from this meta-analysis, these are all studies of water, sanitation, and hygiene that reported participant flows on which we could estimate a all-cause mortality. And we found a large and statistically significant effect on mortality, but very few studies that were sufficiently statistically powered. And unfortunately, none of the studies reported in this participant flow diagram, they're all from epidemiology and public health. None of the studies is from development economics, because those are the authors in development economics haven't reported any of their participant flows. So it's a reason for reporting of participant flows which goes beyond research transparency. It's also it's also an ethical issue that we can actually estimate impacts on all-cause mortality, which is very important for the global burden of disease. Once again, when we looked at this literature, we also found the opposite effects for quasi-experiments versus randomized trials. So we thought that we found potentially bigger effects of high risk of bias. Quasi-experiments, probably due to p-hacking. On the other hand, we found the opposite effect in trials. So high risk of bias trials produced smaller effects. And we suspect this is because of site selection bias. So we have these two opposite effects happening in these types of studies. So sorry, in conclusion, just very quickly, so I would just echo Vivian's one of Vivian's points about its study design is probably the most important factor in determining bias. But we have these, we suspect there are these site selection effects in RCTs, standards for reporting need to be urgently improved in social science trials. And we do plan a project on reporting standards for quasi-experiments. And if you're interested in getting involved in that, please do contact me. We want to build a coalition of researchers and commissioners to exert some change in this field. So thank you very much.
Thank you very much, Hugh. Actually, there is one question for you related to your presentation from Paul Furman. Can you comment on or describe what methods you use for citation and bibliographic tracking in the systematic review that you noted?
Sorry. Yes, sure. So thanks, Paul. Yes, so we used a mixture of citation tracing. So in other words, and what we find we find this in a lot of synthesis research, particularly in the social sciences, where article indexing is just very bad. It's a lot worse. You may think you've got it bad in health. Well, you should come to the social sciences. Very bad. And so what we do is a mixture of both forward and backward citation tracing. So basically the bibliographic back referencing is where we're going through the reference lists of papers that we've identified to include in the review. The forward citation tracing is just, you can do it in Google Scholar. You find your paper and then you go through all the references that have cited that paper. But we also contacted authors. So there are about 150. We estimated when we did this. This was 4 or 5 years ago that we did the searches. We estimated there were about 150 internal replication studies, and maybe 8 to 10 systematic reviews or meta-analyses on this topic. So we were contacting people to identify potential studies that we've missed in the searches. Did that clarify?
Yes, I think so. And there was another request from the audience. If Vivian and you, Hugh, maybe could share with us so that we can share with all the participants afterwards the references you used in your presentations. So thank you very much for a very nice and interesting introduction to our coming discussion. So first I will introduce the panel we have here. We have Karen Robinson, professor from Johns Hopkins in the United States and from Western Norway University of Applied Sciences in Norway. We have Vivian Welch, associate professor at University of Ottawa in Canada and editor-in-chief of Campbell Collaboration. And we have Malgorzata Bala, professor at the Jagiellonian University in Krakow, Poland. So please, everyone, now we have both the two speakers and we have the three panelists here. And there is actually maybe one more question here. Yeah, no, that was just an answer.
Can I ask?
Yeah.
Sorry, just to get us started, I just wanted to ask a follow-up question for Hugh. I think you sort of answered it, but do studies of internal replication actually call themselves that? So why I'm asking is we do a number of meta-research questions, and we look for studies that are meta-research studies, and they don't call themselves that. So I'm wondering if it's the same, if you run across the same issue with internal replication studies, do they actually call themselves that? How do you identify them?
Yes, well, Vivian has quite a good paper on this. So they use a different kind of terminology. So Vivian may be the best person to speak about this, but you hear internal replication study, within study comparison. Are there any more, Vivian? I mean, yeah.
Sometimes they're referred to as design replication studies. Yeah, so actually the way that Peter and I got into the work on replication was that we were working on writing the theory for what's required for a high quality within study or design replication study. And then we realized, oh, replication, this potentially could generalize to other sort of ways of thinking about replication. But yeah, are there other names, Karen, that your field uses?
Well, we don't. I guess that's part of the reason why I'm asking is that I don't think we really use the same terminology. So one of the things that was fascinating to me, I'm interested to hear Malgorzata and Vivian's, is that in some ways it's very similar issues, and in other ways it was like, well, when you're talking, it's like, is that the same concept we have, or is it actually in fact something different? So it's hard. It was hard for me to figure out whether it's a semantics difference or a real difference.
Yeah. I mean, the other type of design I see much more in the medical literature than I see in the social sciences are doubly randomized designs, often to understand preference effects. So basically people get randomized into an RCT arm or a quasi-experimental arm. And the goal is to be able to examine if people have choices too about what, how does that affect the sort of the intervention effect estimates.
Yeah, no, that's absolutely right. And so you might say that possibly in the medical literature, the internal replication studies are more likely to be prospectively designed through this doubly randomized approach rather than, and perhaps certainly in the literature we reviewed in development economics, I think most of the studies were retrospectively designed. So they use what Vivian calls a synthetic design. Yeah.
Yes. I think that the interesting part here is really a situation, as I can see it, make it very simple, that it seems that in social science, the replication is a problem because it's we're lacking validity due to replication. In health, it is actually because of replication, we lack validation, valid studies, so to speak. A lot of unnecessary and invaluable studies are maybe being published. And I think that one of the questions that is really at least important in that respect is, what do we need? How much do we need to say now we know? Should we have 10 studies with 100 in each? Should it be, as you have put, Vivian and Hugh, that we should have a certain aspects of the biases that we should have looked into, that is really making it clear that now we have some sort of a certainty? Or what do you think, the panel and the speakers as well, what do you think is the right way to go? When is replication too little or too much?
Can I follow up
Yeah.
Can I follow up? So I'm so curious about this, the too much replication, because this is, well, in education, they say we don't have a replication crisis because nobody ever does replication, or they don't recognize their studies as replication studies. So I'm very fascinated by this. So can you describe like what happens here? So is it that basically the same an investigator will recruit a sample of participants, and they will conduct the study using the same protocol on the participants, but obviously there's going to be variations in participant characteristics from sample to sample, right, by noise, but also systematic differences as well. And so the idea here is that you have a bunch of sort of independent investigators that are working together, that are not working together, they're working separately, that are recruiting the same or participants that may or may not be similar to each other, and they're running the study, and then they publish their result, and there you go. And that is the thing that feels like, is this really the best way for people to be using their resources to conduct their study, or to sort of contribute to the evidence base? Is that the issue that I'm describing that is happening here?
Yeah, exactly.
I mean, to put it very succinctly, when does replication become duplication?
Yeah, okay. So and then do they always produce the same result? Do they get the same findings? Do they get the same effects? Depends, right?
Yeah, no, and that, I mean, that's, I mean, you touched on it as well. I mean, in the meta-analysis, you might use meta-regression to see why things might be different.
Yeah.
But at some point, like, for example, we might use cumulative meta-analysis to show that, you know, back in 2000, we had an effect, and all the trials done after that showed when you do cumulative meta-analysis, so ordering them by date, all the subsequent trials didn't change your answer. So we would say those were duplicate as opposed to replications, I guess, if we're using that language. But how do you know in social science, is there a way to know, or is there a concept of the redundant or duplicate studies in social science, I guess is the question.
I mean, for us who work in school systems, the challenge is that there's so many different factors that go into play, and each study itself is often so expensive, right? If we're testing a reading intervention, everything from the reading intervention to the control condition to the dosage of, to who delivers the reading intervention, and in the United States, there's tremendous variation across schools. So we do not have issues like that. That's not, that wouldn't occur. But I think where the applicable issue it would be in education would be in special education. So for example, it's not uncommon in special education to do these N of 1 studies where you do repeated measures, you give somebody an intervention, you look at the change basically in outcomes after the intervention. And it's not uncommon because for special education, it's a very specific target population. So it's usually a low incidence group that it's hard to recruit a large sample size, right? So they do sort of these N of 1 studies. And it's not uncommon, for example, like graduate students to go for their dissertations or for their master's thesis to go and do sort of N of 1 or N of 3 types of studies where they do this. And so then the question has become, well, what do we do with sort of this evidence, right? And so I wonder if maybe this is the flip side. Your duplicate question is the flip side of our question, which is that it's about this idea of organizing studies prospectively, right? Like that somehow there's this recognition that we are not sort of allocating our resources in the right way for planning sort of prospective studies. For us in education, actually, it's too few studies, or maybe not the right studies and the right parts of the response surface that we want to actually observe data on. In your case, maybe like there are too many effects of on maybe one cell, but like the idea here is that there may actually be interest in thinking about variation across sort of different types of characteristics or implementations that you might be interested in, but thinking prospectively about designing a whole series of studies that people could sort of then take up those protocols might be a more efficient way of sort of approaching the problem.
Yeah, I was actually, when you were showing your response grid, I think that's what you called it, yeah?
Mm-hmm. Yeah.
That in some ways, I wondered if you could sort of use it the same way we would maybe use cumulative meta-analysis or other things that, you know, once you have all those cells filled in, is there a need to do another study to address that cell, for example? Is that a way to think about it?
Yeah, I mean, I think that's another, that's another way to sort of think about where, you know, like we were always envisioning for using it when data was too sparse, but I think you could also use like, here's what the evidence base sort of has uncovered sort of questions or answers to sort of questions, and here's where we have sparsenesses of data. So if you had to go sort of design a study, like this is the area that would probably contribute to the knowledge base the most. But, you know, I think the idea here is to sort of prospectively, like having a consensus panel in advance to sort of describe what are the boundaries of that surface that that feel important for being able to sort of plan these sets of studies. And then people would have some sort of guidance of what to vary.
You know, when we in health are discussing this issue here with duplication, replication, I think at least that it is a question of the generalizability or the applicability of the study. And if you think of just one study, that's at least how I think about it. If you have just one study, you have too many biases in relation to this. So in that way, I think we need more studies to you know, cover the different biases, so we can say what is actually going on here before we are able to make a full generalizability or applicability of the results. While I think to understand that, at least what you were showing, Vivian, that one study is, you know, it becomes more local. So I'm afraid of the generalizability of the study if you only have one.
Vivian Welch, Vivian, I was going to say W, but that doesn't help us, does it? you just wrote a comment, and I wondered if you wanted to share that with the group. Yeah,
please raise your hand, because we can't keep track of the talking, and so please raise your hand if you would like to say something. That's the best. Yeah.
Yeah. Okay.
Vivian.
Sure. I mean, I just wanted to echo what Vivian Wong was saying, that, I mean, in Campbell, so we publish systematic reviews in the social sectors, and we sometimes have very large reviews, you know, over 50 studies in the education sector that Vivian Wong is familiar with. And I don't think I've ever seen a cumulative meta-analysis like you're talking about, Karen, because there's so many factors that vary in these studies in terms of the setting, the populations, the types of students, components of the interventions, that in fact each study could be testing a different, you know, aspect of that, so they're not, I would say, viewed as duplicative. So yeah, I think there are some interesting differences between health and social science in that respect.
Yeah, Hugh?
Oh yes.
Please.
So
yeah.
I want to respond to Vivian's comment, because this is really interesting. And when she's quite right, I've never seen a Campbell review, or I'm not sure I've seen even a meta-analysis in social science which has ever presented a cumulative meta-analysis. And I wonder, Vivian, if you know, we don't really, and maybe for the reasons that you're indicating, that we have so much heterogeneity of treatments, of populations, that we, you know, this notion of equipoise is not something which can be so universally, like a universal equipoise for a particular topic. Maybe that isn't something that is a relevant concept in social science, but equipoise still is something important, and trials still get done, and we do find that there are. So I'm thinking of in development economic in the international development field, there's a type of study which has been promoted by the World Bank called community-driven development. It used to be called social funds. They were it's a way of engendering a kind of bottom-up development. You give communities some money and you help them to prioritize and design their own projects. And one of the key outcomes that was supposed to address, that the World Bank wanted to address, was related to social cohesion. And all of the studies that have measured all of the randomized trials and quasi-experiments that measured social cohesion, not a single one of those studies has found an effect. And in fact, when we look at the pooled effect, it's just like perfectly centered on zero. There is just no effect of that. I mean, that's not to say the intervention doesn't work, because they do build water supplies and they build schools, but there's no effect on social cohesion. So certainly there's no need for more studies of community-driven development that measure social cohesion, and that would be an example of equipoise. And I imagine if we saw a cumulative meta-analysis, you'd also see that very clearly. So I wonder if, Vivian, what a useful project might be done with the Campbell reviews is to retrospectively construct these cumulative meta-analyses and see if we can answer these questions about equipoise.
Malgorzata?
Thank you, Hans. I wanted to comment because I like the grid Vivian Wong presented, the generalizability, and I was wondering, because this grid can also and should also be used to plan the new studies, right? So this could be like a partly the evidence-based research approach in this, because we are using the currently existing evidence in order to plan for future studies. But it's like a different approach, I guess. Yeah.
I mean, a common question that folks ask usually is, for example, like, how is this different from meta-analysis, right? And I say, actually, this is like, basically, when you get the data, when you move into the analysis framework, we approach it the same way as you would a meta-analysis, right? The only sort of the addition that we're thinking here, or the complement that we're thinking here, is adding the design-based approach to thinking about prospective, what will eventually become a meta-analysis design, right? And we think that, in some ways, there are all sorts of reasons for bias and interpretation that having design-based approaches is useful. Also for reporting, right? How many times have folks done meta-analysis where it feels impossible to be able to know exactly what the outcome measures were, and so having ways to sort of not only plan the effect grid in advance, but to also be able to provide protocols on common measures that all the studies who are going to partake in this are going to use, common fidelity measures that we're going to all sort of use. And, I think would also just be helpful in terms of improving the quality of the meta-analysis that we get on the outside as well too. So that's basically our idea that sort of dovetails, I think, into the reporting things that Hugh had sort of talked about earlier, and helping sort of make the interpretation of the analysis of the synthesis results a bit easier at the back end.
Yeah, so that dovetails well into the question I wanted to ask you, both of you, and because I think you both touched on it. It sounded to me that one of the issues, or one of the key issues, is both a complete and perhaps a common definition or description of the estimate, whether it's a causal estimate, I think as you called it, Vivian, or a treatment effect estimate that I think Hugh said. So I wonder, and I mean, we see this in health research as well, which is why things like COMET, outcome sets have been developed. I wonder if you could speak to any efforts that you are aware of to have something similar to COMET, or if that's even possible, you think, or how can within social science there be encouragement to have complete and perhaps as maybe standard or common definitions of the estimates to help do what you're saying.
Well, I'll just go ahead and speak. In education, I think there are sort of efforts underway to sort of think it. I've been a part of several working groups, and for example, the Gates Foundation is trying to fund an effort to do this. I think the group that I was a part of was using the PICO standards in health, right, as a way for to standardize people's reporting of participants, interventions. I forget what the C and the O stand, but there are efforts. I think it's been challenging in education because, like, once you get a group of social scientists in the room and you ask them what should we be collecting common measures of, it very quickly sort of balloons out. But in this way, I always sort of think like, to me, I think the way to sort of really prioritize is that it's about the moderators, right? Like, at the end of the day, if the goal is to generalize, we really care about collecting information and characterizing information on factors that we really hypothetically think about sort of moderation, right? Or that we have hypotheses around moderation, right? If treatment effects are constant for everybody, right, then it doesn't matter. Honestly, it doesn't matter what the sample is. It doesn't matter what the setting is. It doesn't matter what the dosage is. Like, we have one treatment effect. But it matters a lot if we think that treatment effects vary. And in education, we don't really have a handle on how much variation there is in effects. What's the theory informing it? So, and that is really that's the linchpin for sort of evidence-based decision-making, because people want to know what works for me under my context.
I just would mention that Malgorzata have an urgent thing to do, so you are leaving shortly. Do you have a last comment or something like that?
Can I have just one more question? Because what I was wondering, in education, the interventions are usually complex interventions, right? That's why so much context is important. And it's similar in health sciences, in public health. But I run into problems. So the question is whether it's because you talked about fidelity and how good, fidel, how do you say it as adjective? How well the intervention is reproduced, right? And in health sciences, I think we still have problems with that, with complex intervention, that we don't really know what is in fact within that intervention. We have checklists somehow trying to improve this by good reporting, but how do you deal with that in other sciences? Thank you.
So maybe
do you want to go and talk about it? Yeah.
Yes, Malgorzata. So maybe I can talk about it. So we actually we do so. Latter day risk of bias tools include do attempt to usually have questions about implementation fidelity to measure the fidelity. I'm also not sure what the adjective is. I often wonder what the adjective is for infidelity, but so in these circumstances, it's recognized that it is really important, and indeed those findings that I was presenting about the opposite direction of effect for high risk of bias, RCTs and quasi experiments, a lot of that is probably about implementation fidelity. It's probably that the higher confidence RCTs are better designed and better implemented. So and certainly in the newest tools for risk of bias assessments, some measure of implementation fidelity is always included, even though traditionally we might have thought of those as being more about construct validity than internal validity. And there are even specific tools to look at in implementation fidelity, where the entire tool isn't about risk of bias, it's about implementation fidelity. There was a project that was led by, I think it was Paul Montgomery in about 10 years ago. Sorry, Hans, go ahead.
And actually, I think what we are discussing here is really interesting, that I had noticed that in health, people are talking more and more about the context, more and more about more factors. And to my understanding, and please if I'm wrong in my piece about Sophie and Wong are mentioning it, for example, it's very clear to me that it is so important for social scientists to really incorporate all the different factors that could have an influence. So maybe if I should just paraphrase what we are talking about, artificial intelligence, do you think that we are hallucinating in medical health because we are thinking that we can combine and even make the cumulative meta-analysis and say, now we know? Or do you think that maybe there is a pragmatic approach to this? What do you think of that, Vivian and Hugh?
So maybe I can respond first. No, I don't think you're hallucinating. And but there is this notion of lumpers and splitters, and the lumpers are the people
exactly.
And I'm quite a lumper myself. I actually think that, and because Vivian says it's all about the moderator effects, and in my kind of worldview, as long as we collect the information about the moderators, which kind of links back to having good reporting standards where the moderators are reported, so whether that's some kind of extension to consort for social sciences, where we can explain the moderator effects, which means that we can say how generalizable the findings are. So, and I actually think it's I think it's a misconception about meta-analysis that all we're interested in is estimating a pooled effect, but if that's across such different contexts that it's not actually sensible to do. I think that's actually a big misconception about meta-analysis, because we're interested in the average. We're also interested in explaining the heterogeneity.
Exactly. Very good point. Vivian Wong, please.
Yeah, I mean, I guess I agree with that. I would say that the only thing I would add is that, I mean, I think there's two questions, right? There's what Hugh and what we've talked about previously about, it depends on the extent to which we think there is effect moderation. And I will say in education, people in education have really strong hypotheses about this, right? Some folks think it's basically constant treatment effects and that effect heterogeneity is mostly noise. Others have really strong feelings that it's all context dependent. It's so context dependent that generalization is not even a worthy goal, right? That these are people that are just like, we should be studying qualitatively what happens in specific schools. That's what matters. I would say the policy interest, it also matters for the causal estimate of interest, right? So in the United States, our education system, actually all of our systems, are so decentralized that we have very few policies and decisions that are affected at the national level. And so a average treatment effect that is nationally representative is maybe sort of sounds helpful from a theoretical standpoint, but in practice, that is not how decisions get made in the United States about, and so that's not the relevant causal estimates for the decision makers, right? The decision makers are interested in the causal estimates that pertain to their population of interest. So what I would say here is that there's the scientific question about how much moderation there is and how much constant effects are, and that is entirely dependent on what the intervention itself is. And then the other, it has to do with the policy question of what's the estimate of interest for making inferences.
If I could just follow up on something you said, who are the decision makers and how do you involve them, like the end users and decision makers, in the design of the studies, or do you? How do you?
Well, this is an area that is very new for me because I'm, as a quantitative methods person and a stats person. And so, so we've, over the last 10 years, I would say, at least in education, right, we've thought a lot about having these policy practice partnerships, right, where researchers work collaboratively with policymakers to help sort of identify questions that matter to them, as well as translate findings to them. But the effect grid that we sort of talked about sort of suggested, or that we were thinking about, sort of suggested that we needed new ways for thinking about how to plan studies together, right? And being able to just, because if we're trying to sort of think about how can we plan a series of studies that would be of interest to the field, right? Then this is going to be a more successful effort if we can have sort of greater buy-in in the planning stage. So I've been learning about participatory methods, which I think are sometimes used in public health, right? So public health actually has a longer history in this than us. And I've been learning about sort of Delphi sort of methods for obtaining consensus on identifying the moderators of interest. So that to me that's an area in process that we've been learning about. But so if you guys have ideas from other fields, please share.
Maybe can I add?
Do I have a comment?
Yeah.
Yes, please.
And indeed, there are. It's increasingly recognized that in good quality research, we don't want to just disseminate findings. We want to involve decision makers from the outset. So in the way that Vivian was mentioning, and why would you do that? Well, it's partly to get the questions right. So we actually ask questions and we test them with empirically questions which decision makers actually want answers to, as well. And that's not always a question about the average treatment effect. And it also helps to kind of build ownership amongst those communities, and avoid I can't think of the right word, but avoid a circumstance where decision makers might be blindsided by a very negative finding, because we don't always, of course, find positive effects. And so there are.
Would that mean that? Yeah, sorry. Please continue. No, I was just thinking that maybe the evidence-based research concept is really promoting the idea of valuable research, not just a matter of getting published as much as possible, but maybe less research, better research, and research done for the right reasons, as Doug Altman was saying 30 years ago, actually. So what we are thinking is that we should base our new study on the validity, both the internal, the construct, and the external validity of existing studies dealing with the somewhat similar questions. We should add to that the end users' perspectives, concerns, experiences, meaning those who will either use the study you're planning or will be affected, for example, students in teaching by that, and divided by the cost of being involved in the study. That's what we think is the valuable research, and we will really promote that this kind of research should be, we should look more into that. What do you think of that?
Okay, that's a very complicated question.
Okay, sorry.
But no, that's fine. Just because, I mean, the big issue, of course, is that we don't want, just because like a decision maker doesn't agree with the findings of a research, that should then factor into whether, you know, that research should be done, right?
Exactly.
But at least by involving the decision maker. So at Campbell, Campbell uses, for example, stakeholder advisory groups. So in a Campbell review, there should be a group of end users that are convened at the start of the project, and they get to have a say in, they don't get to have a say in the methods that are used, but they get to have a say in the, they don't get to have a say in how rigorously the methods are conducted, but they get to have a say in what types of questions are asked at the start of the review project. But you know, that approach actually comes from primary research, so studies done on of primary research, RCTs and other studies, where it was quite seen. I can't remember, maybe at least 10 years ago, it was seen that if you don't involve end users from the outset and you kind of blindside them, that you basically, you alienate your end users, and then you don't get evidence-based, evidence-informed policy, basically. So, and yes, like as Vivian said, they might use a participatory approaches, or you might convene a Delphi panel, as Vivian Welch has done on a number of different projects. I did want to. So Karen, I wanted to answer the question that you posed at the start, but perhaps if you've got a follow-on question, then I'll come back to this at the end, whenever that is.
Can I make a comment here? That maybe this is just a question, since you guys may have more experience in this. So I sit on the board of a registry, of an evidence registry, and they sort of shared, even though the registry's been around for a while, only recently have they convened basically a participatory sort of user group to understand how people are using the evidence from the registry. And the registry was really conceived as sort of like a consumer report for different types of education products, right? But it turns out that first, people in the field don't really use the registry very much at all, period. And one of the reasons is because there are very few people, like, for example, teachers really do not have a lot of decision making about what curriculum or interventions they use, right? Somebody purchases it for them, and they have to use it. But when they do use it, they use it for a purpose that was different than how it was conceived, right? Usually they're looking to see, what does the evidence say? And then they kind of reverse engineer or map what they're currently doing, if that's aligned with what is being recommended by the registry, right? So they may not be using the curriculum that was specifically evaluated, but they're looking to see, oh, well, does the curriculum have like phonics instruction? That seems to have this evidence that was supported in this study. So, but as I was listening to this, it really struck me that how folks are using this evidence registry is really different from how the evidence registry was originally conceived, right? Like it was really conceived as a consumer report kind of thing. But it occurs to me that how you might, like, if you knew from the end, at the end, that this is how people were using the evidence, that you might not have developed an evidence registry that really spends 90% of its effort on specifically coding each study to go into so I'm just curious if you guys have had experience with this, like this disconnect between how people use it and then how you set up the registry for it, and if there are alternative ways. That was a very long, meandering question.
You had the.
Oh, I don't know, it's not really answering your question, Vivian. I had a kind of a follow-up to Hans' question and Hugh's response, and that is, you know, the evidence-based research network, which emphasizes basing decisions around research, whether to start a study, how it's designed, and how it's reported within the context, should consider existing evidence. So it's evidence-based. And historically, we have focused on clinical research, and we've been trying to move into other kinds of research. So I guess my general question to you is, how do you see, or do you see, evidence-based research being important in the field of social science, or how would it be different in the field of social science? So I guess as a sort of a general question, following up on what Hans was asking.
Yeah, I mean, it's been complex. And people, I don't know, Hugh, I don't know if you've sort of, this is not my area of research, but it's something that I spend a lot of time agonizing over, because we think about how to produce research that people will find useful. I think what I have been thinking about is that for the end user, there's been a lot of ways that people seem to be interpreting the evidence, right? They do not use it as a consumer report style. There's some mediation that seems to be happening between what their employer tells them to use and who people they seek advice from. And it's almost like they're using the evidence to confirm what they either their priors or what people have told them. And I'm not quite sure what to do with that, to be perfectly honest with you. I do think there's probably a lot of work that could be done in this space. And I don't know, Vivian, Welch, and Hugh, maybe you guys have some additional insight on this.
So would you call that, Vivian, what do we call that? Is that policy-based evidence making? I'm not sure, but
policy-based evidence making.
But what I would say is that it's kind of interesting, because I often think, and I'd be interested if what the Vivians think about this, that we're living through a kind of an evidence-based revolution really in the social field. Maybe you don't so much in education, because education is a bit more advanced than the other kind of social areas, but often we think that, and you'll often hear that, well, in social science we're about 30 years behind health, that basically, you know, everything that you've been doing in health, it's kind of interesting because it's a bit of a misnomer actually, because meta-analysis originated in the US.
Exactly. That's what I was thinking when you said that.
But it was really taken up by the health community under the auspices of Cochrane. And so, you know, so the extent to which like evidence-informed decision making is taken in health is just vastly exceeds that in the social areas. So we are kind of we have a lot, we can learn a lot from the experience in health. So it's very nice, and in some areas we are 30 years behind health. For example, the reporting of participant flows in trials. I mean, maybe in education, trials get reported with consorts, but they don't appear to in development economics, for example. But at the same time, and I mean, maybe this comes back to Karen's question at the start. Karen asked, when is replication too little or too much? And but there are some areas where social scientists are doing work where I think health people could draw on these, and indeed in some cases are. The example being these internal replication studies that Vivian has so eloquently devoted quite a big part of her career to, as I understand it. And the advantage of these studies is that they actually give us a way of testing. They give us a way of verifying our risk of bias tools empirically. And so we know that we have risk of bias tools that, and again, they largely come from health, but they're largely theoretically derived and they're not empirically validated. And so one area where I think that the health people can actually draw on the experiences of social science is through these internal replication studies and syntheses of these internal replication studies to develop better risk of bias tools, more appropriate risk of bias tools.
Don't repeat our mistakes in health. Don't do that. And I can, as an answer to your question, Vivian Wong, that when it relates to the involvement or the engagement or the incorporation of end users' experiences, concerns, perspectives, values, preferences, it has become, I don't know what we should call it, but it has become something you're just doing because you have to. And in many cases, it's really doesn't make a difference. It's just something to get funded. And what we are doing in the evidence-based research community is really to state, we need to use a scientific approach when we are incorporating end users' perspectives. That's really what we are aiming for. So yeah. And we would love, honestly, we would really love to have a collaboration with social science, because we think, just as you put it, Hugh, I think we can learn so much in how to really deal with this, not just repeating the same study again and again and again. And we have so many cases that people are doing that just to get published. So we need really to collaborate on that. So I really hope that we, by having this seminar, maybe could meet again and discuss that. That could be very engaging and exciting for us to have a talk with you about this, because I think there is a lot of common areas that where we could meet and discuss. Maybe some semantics a little different. Don't think that matters that much. No.
Yeah. I mean, I think what I'll say is that when I started in graduate school, IES was just launched, right, in the United States funding agency. And the idea was to launch this agency to be able to replicate identifying effective interventions in education that was modeled after the FDA drug, approval process, right? And the idea would be that you would move along this sort of linear function, and that we would be able to identify in this registry what interventions have worked. And it's been 20 years since that process was introduced to the field, and in some ways it was incredible in that it has really moved education much more towards rigorous evidence-based evaluations. On the other hand, we're finding it's not really meeting the practitioners' needs, in that I think people have strong hypotheses that the context and the implementation really matters so much in this, that this question we've been struggling with as a field. How do we engage with not just what works, but what works under what conditions and for whom? And sort of how do we do that in sort of a systematic way? So that's so I think that's definitely an area where our fields can kind of move forward in thinking about that together.
In fact, can I just follow it? So Vivian,
Yes.
Vivian talked about the PICOS acronym, and they said the population intervention, comparison, outcome study design, and it's the C bit, which is the comparison, which can be so important for what the size of the effect is that is estimated. So, you know, when we know that we would expect that if we have a study where the counterfactual receives nothing, we might expect, for an efficacious treatment, we would expect a bigger effect than a situation where the counterfactual gets something different, gets an alternative. And this is indeed this is why we specify the C in our systematic reviews, and it's why we test for moderator effects according to the C. And generally, in most of the reviews I've been involved in, it's the counterfactual which is one of the biggest determinants of the effect size. And so I think we need we need to define much better not just what our interventions are, but also in our counterfactual, our comparison group conditions, and this is something which we should work on in our consort extensions for social science.
This has been very, very, very exciting, interesting, and thank you so much for joining, and thank you, Vivian Welch, for helping us to get acquainted with those two very good people here. We should definitely meet again in one or another way. So thank you, all of you participating in this, all the listeners and participants in the seminar, and hopefully see you all again soon in one or another way.
And feel better, Hugh. Thank you.
Yeah, feel better.
Yes.
Yeah.
Yes. Thank you.
Bye. Thanks.
Bye.
Again.
Thank you so much. Thank you.
Thank you.
Have a good day.
Bye.
Bye.
Thanks.
There's another